Posso andare? Ok, è partito. Buon congresso, buon talk a tutti.
Grazie.
Okay. Good morning, everybody. We resume today with a scientific seminar that accompanied us in the first month of the year. Today, I am pleased to introduce a colleague, Dottoressa Francesca Giuntini, whom I had the pleasure of meeting for the first time at the Congress on the Photodynamic organized in Bressanone in 2012, and with whom an interesting collaboration has been born over the years. Francesca Giuntini obtained her degree in medicinal chemistry and pharmaceutical technologies from the University of Florence and her PhD with Professor Giulio Iori. After the PhD, Francesca worked for a year at Molteni Farmaceutica in Florence. Then Francesca held a postdoctoral position at the University of Aveiro, University of Bath, and at the end at the University of Hull. Following which she took up her appointment as a lecturer of medicinal chemistry in the School of Pharmacy and Biomolecular Science at Liverpool John Moores University, where she is currently a reader in photoactive therapeutics. Francesca's interests focus on the development of novel biomedical applications of photosensitizing processes. She is interested in designing bespoke porphyrins for this application, and she is especially attracted by the association of porphyrin with smart molecules. So now I'm in a pleasure to introduce Francesca.
Ok, hello. Ho deciso che la prima slide, la presentazione è in inglese, però mi presento anche da sola in italiano perché mi faceva strano parlare in inglese in un congresso della SIF. Però quindi. Allora, prima di tutto grazie Enrico per avermi per la presentazione. Io sono Francesca, lavoro come professore associato alla Scuola di Farmacia e Scienze Biomolecolari dell'Università Liverpool John Moores a Liverpool. Questo qui è l'email, se qualcuno di voi fosse curioso a proposito delle cose che dico e mi volesse fare domande dopo, volesse entrare in contatto, fate pure. Allora, qui c'è una porfirina perché, come ha detto Enrico, io mi sono formata come chimico farmaceutico a Firenze e da allora ho incontrato, mi sono innamorata perdutamente di queste molecole qui e mi piace pensare di essere un chimico farmaceutico che lavora sulle porfirine. Liverpool, noi siamo qui più o meno. Questa è Liverpool vista dall'altro lato del fiume in una notte inusualmente chiara e venite tutti, non venite tutti insieme se non c'è posto, ma venite a trovarmi perché la città è bellissima, la gente è parecchio ganza, come si dice, probabilmente avete capito che non sono proprio originaria di Liverpool, vi lascio immaginare di dove sono. E c'è tante cose da fare, si può andare a vedere i Beatles, si può andare a vedere il calcio, si può andare nei pub, eccetera, eccetera. Quindi venite tutti che vi aspetto. And now we swap to English. So let me just do one thing before lo vedete in mouse, posso fare con la freccia del mouse invece che con il pointer?
Sì, vediamo la freccia anche del mouse.
Ok, perché altrimenti non mi riesce. Right, swap to English now. So this is an outlook of what I'm going to talk to you about. So I'm going to start from why it's a good idea to consider porphyrins as agent for therapy, as drugs. And I'm going to cover how, okay, they're good, but they can be improved and the ways to make porphyrins better drugs. And in particular, how to do it by using a particular species, particular family of compounds, which are peptides. And I'll show you some applications. And in particular, I'm going to focus on projects that I've been involved in with since I started working in this field. So how we can use them to improve cell access and how we can have favorable intracellular trafficking, so intracellular localization, how we can target bacteria and how we can use them to formulate porphyrin better. Now, all of these, why porphyrin should be used as therapeutic agent cannot be described unless we talk about photodynamic therapy. I'm fully aware that in the group of people I'm talking to, people know better than me what PDT is, but just for the sake of the discussion and for the people that might not know enough of that, I'll quickly cover it. So photodynamic therapy is a treatment that is done with drugs and with light, and it entails basically the administration of a drug that we call photosensitizer. We wait for a certain amount of time for the drug to localize, if possible, in the interested tissue where the disease is localized. And then we proceed to the irradiation with visible light, and then ideally the disease, the ailment disappears, the disease is cured. Now, this approach was described a long time ago at the beginning of the last century, and what attracted a lot of attention from the start was that people observed that in the absence of the light, there was no effect. So this, in other words, is intrinsically a very selective treatment. So you only have the action of the drug where you shine the light. And all the diseases that need selectivity, perhaps cancer is the main one. And so this treatment gained a lot of attention as a potential anti-cancer treatment. And indeed, from its inception, from its discovery, it has been tried, tested, and approved in some cases as a treatment against cancer. You have here a list of a few of the tumors that this treatment is used against. But not only tumors, but you can treat pre-cancerous diseases like dysplasias, for example, also age-related macular degeneration, which is a disease that impairs the vision, especially in the elderly, actinic keratosis, infections and inflammation in gum, and so periodontitis, and so on and so forth. So it has been tried for quite a number of diseases. This here is, you can see that the first large clinical trial, which was something that increased the attention of the scientific and medical community for this approach. The first clinical trial was done quite a long time ago in '78, and it was a trial that involved 113 patients with different kind of subcutaneous and cutaneous tumoral lesion. And it was proven that the treatment was overall safe, and interestingly, no tumor was found to be non-responsive. In other words, what it meant that this treatment could hit any tumor, more or less, that it was directed against. Of course, these kind of people went to town with that, and since then there has been a high number of clinical trials, I think 200 since last time I kind of counted, and 200 to the power of lots of fundamental studies and preclinical trials. So it still gathered a lot of attention. This is an article that tells the clinical story of PDT against cancer. It's from a while ago, but it's a very interesting piece of reading. Now, okay, so I told you that we have a photosensitizer light, but what happens behind the scene? You might have seen this probably, so I can't talk about PDT without showing a Jablonski diagram. As I said, the drug here I'm depicting it as a porphyrin. It's a molecule that is able to absorb visible light. Once it does that, it populates the first excited singlet state. From this state, by intersystem crossing, it populates the lowest triplet state. And from there, in the absence of oxygen, the molecule would either relax without emitting radiation or emit light and be phosphorescent, or in the presence of oxygen, it transfers either electrons or energy to molecular oxygen, generating the actuator of this treatment, which are the reactive oxygen species. The main ones for PDT are singlet oxygen and superoxide radical anion. Now, these things, they do what they say on the label, so they're reactive. They don't have a long life in the biological environment because they react literally with anything that they can find within their reach. For example, they can trigger the oxidation of polysaturated fatty acid and therefore potentially damage cell membranes. They can oxidize most of all guanidine, but other nucleic bases as well, therefore damage nucleic acid like DNA or RNA. And they can oxidize some of the side chains of amino acids, thereby bringing damage to proteins, receptors, enzyme, you name it. So they're quite dangerous little species, and it means that photodynamic damage can be done to pretty well any structure inside the cell or around the cells. This slide here shows most of the clinically approved photosensitizers, and you can see that even to people that are less familiar with chemistry, there is a motif that is recurrent. So this large squarish structure that is a porphyrin lot of double bonds conjugated. This is a phthalocyanine, similar but different. This is different, is a hypericin, but they're all large aromatic molecules, and many of them are tetrapyrols, and many of them are porphyrins. Which means that these species, because they are approved photosensitizers, means that they are good photosensitizers. The only guy that sticks out like a thumb in the middle of this is this little guy here, which is neither aromatic nor large planar molecule, but this is aminolevulinic acid, and in fact is a prodrug that in vivo is metabolized to a porphyrin. So again, it's a porphyrin. So porphyrins are good photosensitizers, and in fact, they are not this is not the only reason why they attracted a massive interest from the scientific community. The structure of a porphyrin is so unique.
Allora, se voi lo volete pagare il tampone,
Okay.
a me serve
So it can
comunque un tampone molecolare che costa 70 euro. No, questo qua no, perché costa 70 euro.
Anyway, so as I was saying, nature chose the porphyrin ring moiety to achieve some functions which are linked to life itself. So for example, for us, the animals, a porphyrin is at the basis of the transportation of gases in the blood, like oxygen and CO2. And the audience I'm talking to is the last one that needs to be reminded the importance of chlorophyll, I believe. So anyway, they're known to be pigments of life, and they also attracted interest in other fields than photodynamic therapy. But back to PDT, why are they good photosensitizers? Well, because if you remember this graph I showed you a couple of slides ago, because they're particularly good at doing this kind of trip around the excited states. They're good absorbers of visible light. They can, not all of them, but most of them, they can populate efficiently the triplet state, which is essential to then generate reactive oxygen species, which they do particularly well as well. They have limited dark toxicity, most of them, meaning that, again, the selectivity of the treatment is maintained because no light, no party in a way. Another reason, especially for a medicinal chemist like me, is that I forgot to say that another good feature of porphyrin is that they absorb the right kind of visible light in a way, because you know that different wavelength of light, they penetrate differently in the tissue, better or worse. Now, porphyrin absorb in the blue, so they're good for delivering superficial photodynamic therapy, but they have absorption in the green as well, which allows to go a little bit deeper. Modification of porphyrins can even make them absorb in the red, which is the deepest delivery of the treatment that we can have with visible light. So I was saying, as for a medicinal chemist like myself, one fantastic piece of attraction of porphyrin is that they're chemically accessible. So I can make them, okay? I can make them relatively easily, and not only that, but by changing the nature of the groups that are here that I represented with kind of colored circle, and introducing metals and the kind of metals, I can play with the properties of the molecule and try to get what I want of the porphyrin. So I can tune the properties of the species with the chemistry, which is the paradise for a medicinal chemist. They're good, but they're not perfect, as I said, so we have margin of improvement. What we don't like very much of porphyrin as drug is that they're not exactly water soluble so they're not soluble in the biological environment. And also when they are soluble in water, they tend to aggregate unless you do something to them. And aggregation is something that is not really good for PDT because it changes the behavior of the excited state and the behavior with respect to light. So you still have a molecule that absorbs, but then the translation of that energy into reactive oxygen species is impaired and is not that good anymore. The other thing that is possible to improve because it's not that good is that they lack selectivity for the target tissue. Now, fair enough, we said before that the treatment only works where there is light, but if you have a drug that selectively accumulates in the target tissue, is even better, you have even better selectivity. So people clearly, the scientific community got challenged by these two hurdles, and they went to town to try and overcome these issues. And people started to associate porphyrin to different species to make them behave better in biological environment. Large family of compounds have been made by attaching porphyrins to sugars. And if you know, sugars are very, very water soluble. So attaching a porphyrin to sugar immediately makes it more water soluble. And yes, it has been attached to monosaccharides, disaccharides, oligosaccharides, complex carbohydrates, cyclodextrin, and anything that pretty much was, sorry, got stuck, back on. So yes, they have been attached to species like this. And besides the increase of water solubility, if you pick your carbohydrate in the right way, you can even target particular receptors that maybe are expressed on the surface of a cell that you're trying to target. An example, mannose and some kind of cancer cells. Another approach that has been attempted and is being pursued is the association of porphyrins with nanoparticles. Now, if you Google porphyrins and nanoparticles, your computer is going to suffocate, because could fill big rooms in libraries with articles that deal with this topic. And people have chosen nanoparticles made of different materials like polymers or silica, metals, nanocrystals, you name it, they've used it. And the scientific community was really inventive and creative to associate the porphyrins with these species. So they have encapsulated the porphyrin in the nanoparticles, attach them to the surface, absorb them to the surface, create an electrostatic interaction to make it stick there. Every possible way to incorporate these species was really gained. The advantage of using this approach was that once the porphyrin is in the nanoparticle, it cannot aggregate as much as it does when the nanoparticle is not there. The nanoparticle sometimes protects the porphyrin from the environment. So you might have, I don't know, a porphyrin that attaches to the membrane and doesn't go where you want it to go. And also you can use the nanoparticle as literally a little bus that delivers not only the porphyrin, but other drugs to your site of interest. The third approach, one other approach, which kind of serves as the springboard for me to introduce the topic that I'm going to talk more about, is the use of proteins. So this cartoon here represent an antibody and the porphyrin attached. So antibodies have been used, and of course, you know what attaching a porphyrin to antibody means, that then it will seek actively the antigen that the antibody was trained to recognize. So it's a fantastic way to direct the porphyrin to the target cells or tissue. But also people have used transporting proteins, like, for example, albumin. So you can load the albumin with a lot of porphyrins and exploit, for example, the tendency of I don't know, cancer cells to the greed of cancer cells for proteins and nutrients to accumulate the species in here. A subfamily, not protein, but shorter protein, are peptides, and this is what I'm going to focus for the rest of the talk. It's a particularly successful and attractive approach to make porphyrins better photosensitizer. Briefly, what are peptides and why do we like them better than proteins? In some cases, well, they are short sequences of amino acid, and by short I mean anything as short as 2 amino acids, that's already a peptide, up to 50. Beyond 50, it's borderline, you really start talking about proteins as well. The beauty of these, again, this is the medicinal chemist talking, is that they're synthetically accessible, so they can be made chemically. Not only that, but the field of peptide chemistry is vast and robust enough that there are a lot of ways to do chemical variation to, I don't know, for example, favor the attachment of porphyrins to peptides, and yeah, chemical modification. The methodology is there and it's robust. They're cheaper than proteins because you need just to pay a person to make the peptide rather than train a cell to produce the protein you want. They have less potential of inducing immunogenicity, and so they're less immunogenic than proteins. And there are tricks that you can do to increase their plasma life. So you by, for example, introducing non-natural residues. With porphyrins, of course, they are good because they increase the water solubility compared to the porphyrin, most of them. They reduce the tendency of the porphyrin to aggregate. So again, 2 ticks. And also you can select your sequence to direct the conjugate wherever you want. So you know that you've got some receptors on the surface of the cell. Peptides can be chosen as ligands for these receptors, or you can increase the penetration of the with suitable sequence of the conjugate in the cell. You can direct to a particular intracellular compartment. So as I wrote here, the sky is the limit. There's quite a lot you can do. Now, how do you attach porphyrins to peptides? Well, that's a exquisitely chemical problem, and you can choose a number of approaches. One, for example, is to attach the porphyrin to groups which are always available on any peptide sequence, give or take, which are the amino group and the carboxylic group. At least you have one of each in every peptide. And of course, it's relatively easy to insert on the porphyrin the complementary function, so the partner functional group that will allow the connection to be made. And you can form an amide or you can form a thiourea. Advantages of these approaches that, as I said, you always have at least one of these species on a peptide, pretty much. But this might pose problem because of the selectivity, because these are rather abundant groups in peptide sequences. So it might mean that you have 2 amino groups or 3 amino groups. And at that point, it's very difficult, say, for example, if you want to attach only one porphyrin, to target that group in particular. But this is a work where this link between a porphyrin and the peptide was made by the group of Céline Frochot in France quite a while back now. They attached this protein to, this porphyrin to EGFR ligand, so the receptor from the endothelial growth factor, and this is a ligand for that. If you don't want to use this approach, you can try to target less abundant residue, but still natural residues. And the king of this residue is cysteine, because cysteine has a sulfhydryl group, a thiol group on the side chain. And you can do fantastic chemistry, as I will show you in a few slides, if you use this group here. And because this group is a favorite in the bioconjugation world, so we know groups that can be installed on the porphyrin to make this ligation particularly smooth. And again, this has been used quite a lot. And the advantages is that this amino acid is perfectly natural, it's cysteine. In some case, tyrosine can be used as well. Slightly different approach, but still natural amino acid. But they're less frequent. So inherently, it might not be there. You might have to add it to the sequence. This is not the end of the world in terms of the feasibility of the peptide, but if your sequence doesn't behave anymore like it was behaving before you were adding the amino acid, you need to check this sort of thing. So it might change the behavior of the peptide. An example of this is a superoxide dismutase porphyrin, mimic porphyrin attached via the thiol to a mitochondrial localizing sequence. Newest kid on the block is the conjugation that relies on the use of non-natural amino acid. And one of these approaches is particularly popular these days because a Nobel Prize has been awarded a couple of weeks ago to the people that kind of applied this approach to cells, in the cells. So this entails, as I said on the introduction, on the peptide, of residues that nature doesn't make. And of course, you have to introduce, as usual, in the porphyrin, the partner functional group to allow the conjugation to take place. Examples of these are the Huisgen reaction, also known as click reaction, Nobel Prize one, Staudinger ligation, a bit more tricky to install this group, but feasible, and the metathesis reaction. Advantages of these is that this is exquisitely they're selective, sorry. So you don't have any other group that behaves like this that nature makes. So there is minimal cross-reactivity. The porphyrins will go where you want them to go, not on other groups. Of course, because it's not natural, you have to find a way to put it in. This is an example of a species, a conjugate made with a derivative of a porphyrin, it's not quite a porphyrin, this one, with a cell-penetrating peptide. Now, once you've chosen your approach, there is another selection, another choice that you need to do. When do you want to do it? Now, you have 2 options. This here is very brief overview of how you assemble, how you make a peptide chemically in a lab. So you take, normally nowadays, a peptide is made on solid phase, which means you take a bead of polystyrene and grow the chain of the peptide on it and then detach it when you're done. So this entails you take your functionalized polystyrene bead, you attach the first amino acid protected to prevent side reaction from taking place. Then you deprotect the first amino acid. You attach the second one. So you have your bead, first amino acid, second amino acid protected, deprotected, attach the third, so on and so forth. You cycle these 3 steps through until you reach the sequence that you want, okay, on the bead. Now, at this point, you can do either you choose to attach the porphyrin while the peptide is still on the resin. And in this case, yes, as I said, you attach the porphyrin, then you detach the conjugate from the polystyrene bead and you obtain the compound you want after this. Or Once you have the peptide on the resin, you detach the peptide and then you perform the conjugation in solution. Which one is best? Well, it depends on the system you've got, because in some cases, for example, this on resin approach has advantages because it's much easier to be selective. There are ways for you to, I don't know, deprotect or make one particular residue reactive. So it's easier to make sure that the porphyrin goes exactly where you want without introducing fancy residues at all. But then on the other hand, if your porphyrin doesn't survive the conditions quite harsh that you have to use to detach the whole conjugate from the resin, then you cannot use that. So it's good that you have these two approaches, okay? This one difficulty of this in solution method is that sometimes the peptide is water soluble and the porphyrin is not. So yeah, you got to kind of see the system you've got to choose for the best. Now, what did we, the kind of a piece of work that we did quite a while back, it was started when I was in Bath in my postdoc. We wanted to explore some kind of bioorthogonal or selective approaches to link porphyrins to peptide in solution. And we decided to explore quite a bit. So first of all, we picked a model sequence. This guy here is 13, if I'm not wrong, peptides, and it's a part of the penetratin, which is a protein that allows the relocation of the HIV virus components in the cell. So it's a viral protein peptide, and we chose these as the photosensitizer. So the first thing, we assembled the peptide on resin, as I showed you before, and then we moved on to inserting on the peptide the function that we wanted to try as conjugation techniques. So in one case, we attached a cysteine, an amino acid, at the end of the peptide, and so cysteine is the amino acid that has the SH. So this one possibility, one of the groups that you can use for conjugation. Then we attached, in another case, still here, a long chain containing an azide for a click as azide alkyne conjugation. Shorter molecule with a triple bond, which is the other version of these. So we thought of swapping the partner of conjugation to see which one worked better. A shorter azide, we're just exploring the chemical space here. And then yet another approach was the one that exploits the carbonyl compound to link to form an oxyamine carbonyl conjugation. Okay, so we had 5 peptides with conjugation handles. Then we moved on to making from the starting porphyrin the partner, the conjugation partners on the porphyrin. So for the thiol, we introduced a maleimide. On this, we linked a bit that then carries a maleimide group. Then for the click reaction, we introduced a triple bond to react on the peptide bearing an azide to react with the peptide bearing a triple bond. And this particular group here, which has a triple bond, it works with azides as well, but whereas these ones requires the addition of copper, this doesn't. And if one day you're thinking about labeling something in a cell, loading the cell with copper is perhaps not the best idea you can have. And lastly, we attach this derivative that is, let me move this. So this derivative here that then is deprotected to form an oxyamine. All of these reactions were carried out in solution, in solvent, in conditions which I would define quite classic for amide formation in organic solvents. Okay, so we had all the partners in place, and we verified that in solution, in the presence of the deprotected peptide, we had the formation of only one selective conjugate. So the porphyrin was linking to the group it was meant to react with and nowhere else, which was good. The same thing for the carbonyl reactive group. It links here in the MSO with a little bit of trifluoroacetic acid. Good yield as well, good, so good outcome, single product. The click reaction worked very well. It's click overnight, so it's not that click. I was expecting them, I remember, to be quicker, but never mind. And as you see here, the one that doesn't require the presence of copper, you just have to put the two things together that will click. In the case of the terminal, so that the triple bond, terminal triple bond, you have to add copper. And if you have to add copper, it's a good idea to introduce zinc in the porphyrin, because otherwise copper will go in and then your porphyrin is not going to behave anymore as a photosensitizer. but it's easy to put zinc in and eventually, if you so wish, to take it out without scrambling or ruining the peptide at all. Now we had a lot of conjugates. What we did with that, as I said, the sequence that we chose is a cell-penetrating sequence. So we tried to see whether it was cell-penetrating or not. So did the peptide behave like a cell-penetrating peptide, and did the porphyrin still behave as a photosensitizer? So we exposed cells, rat fibrosarcoma cell line to the conjugate. And after 24 hours, we went and took the micrographs, and you can see in this A here, you see the fluorescence of the porphyrin, which it's inside the cells. Here, so the conjugate is you showed that it's inside the cell. Here in the middle is the same shot, but done with lysotracker. So this is a probe that shows you where the lysosomes are. And if you merge the two images, we could show that these are the two are superimposed, meaning that the cell-penetrating peptide was still cell-penetrating, and it carried the porphyrin inside. And we think we kind of showed with these images that it goes in via endocytosis and then it ends in the endosomes. Does the porphyrin work as a photosensitizer still? Yes, it does. And this is why we can say that. So here you see this is no conjugate, and this is increasing concentration of conjugates. The blue bars are no irradiation, and then the other bars are 1, 3, and 5 minutes irradiation. So you can see that the more light you give and the higher concentration you give, the viability of the cell is reduced. So yes, the answer to the question, does the porphyrin still behave like a photosensitizer, is yes. Now, another thing that you can do with these, with conjugates like this is slightly different. The cysteine was attached in a different place, but anyway, same conjugation approach. We use this conjugate as an antibacterial photosensitizer. Now, I don't have the time here to go into the many advantages of using photodynamic therapy as an antimicrobial treatment, but I still think it's not used enough and not exploited enough as it should be. Because, I mean, it has been shown that it's promising because it has very little potential to induce resistance, which is a problem of many other antimicrobial agents. Why these conjugates against the bacteria? Because it is known that positive charges increase the ability of any drug to get into the bacteria, or at least associated to the bacteria. And here in the sequence, I indicated in yellow the residues that bear a positive charge on the side chain. So this is a highly positively charged species. We took these conjugates and tested it against 4 bacteria, 2 gram positive, 2 gram negative. And you can see this is the dose response. So this is the viability of bacteria in a way. So there is, of course, if you increase the concentration, 1, I think is micromolar, this, you can see some toxicity in the dark, but switching on the light, the toxicity is higher. Most of all is higher in particularly nasty bugs like Pseudomonas aeruginosa. This is the graph that shows the light responsiveness. So the same dose, I think this is, it was 0.5%. And if you increase the exposure to light by increasing the time of illumination, then the more bacteria die, which is logic. But in science, things are logic only after you've proven them. We wanted to see whether this was for any reason, whether we could see the fluorescence of the conjugate associated to the bacteria. And yes, these are the graphs that show that yes, that's the case. So a higher concentration of the conjugate increases the fluorescence emission from the cells. So yeah, these species gets in associates to the bacteria. Of course, this is something that we did with porphyrins, peptide, antimicrobial peptide, and bugs. This is a massive family. So again, if you Google peptide-based antimicrobial porphyrin, you're going to be inundated again. Another 2 examples, this is porphyrin conjugated to an antimicrobial peptide polymyxin B, and porphyrin and apidacin conjugates. Again, these are antimicrobial peptides. Massive family, lot of peptide tried, variety of photosensitizer, massive range of microbes that were tested as their resistance was tested to these species. It's promising. I hope people, and I hope some of you will carry on this work as well. I want to do it. Once recently we had the chance to explore another, well, bioorthogonal, almost bioorthogonal approach, selective approach, I would say. Recently we were trying to exploit a reaction that is known, whereby a thiol displaces an aromatic fluorine. And so this is a reaction that happens really easily in really smooth conditions. So we say, can we do this in porphyrins? We revisited this reaction because it is known. There's a lot of compounds, many compounds that have been made by exploiting reactions that replace that where the para-fluorine in this porphyrin and its derivative was exploited to make different kind of compounds. And this is reviewed, it has been reviewed quite a few years ago, but people have displaced this fluorine to attach sugars, to attach nucleic bases, polymers, peptides, many species. With nitrogen nucleophile, oxygen nucleophiles, and not so much sulfur nucleophile. But because we were looking at this particular reaction, we said, okay, let's see if we can develop a smooth, kind method, gentle conditions to attach the protected peptide to these porphyrins. Turns out that yes, it's possible. So this is an example. This is a short sequence of the peptide that has a sulfide, so a thiol. And in half an hour, in very, very mild condition, in the presence of cesium carbonate as a base, we have full substitution all around the porphyrin. It's in fact is so good that it's difficult to control it and have only one. We can do a little bit more with this. We can, if you take a sequence of peptides that have 2 cysteine, we can staple the peptide across the porphyrin or around the porphyrin. It depends on the length. And we tried these. We took the porphyrin and we took a sequence, a peptide sequence that is called SPACE. And this is a cell-penetrating peptide that works on keratinocytes, so skin cells. We made this compound, we verified that the porphyrin still spectrally behaves like a porphyrin, so it absorbs and emits like a porphyrin. The conjugate in itself, if you don't switch on the light, it doesn't do anything bad to the cells. This is a keratinocyte cell line, this is a lung cancer cell line, so it's non-toxic to either of them. And then we could see that, okay, so the porphyrin behaves like a porphyrin and the peptide, cell-penetrating peptide behaves like a cell-penetrating peptide. These are the keratinocyte where you see a nuclear staining and the kind of punctuate distribution in the cytoplasm, perhaps indicating lysosomal accumulation. These, I apologize, the red, these red blobs are the nuclei. They're not stained with the porphyrin. It's the nuclear stain that shines for some reason here. So that's a bit of a wonky picture, but sorry. And here is the lung cell where you see the nuclear stain, but not the conjugate. So it just gets in the keratinocytes. So this is a nice reaction. We want to do more with that. Now, another application of conjugates of peptides with porphyrin can be used for drug delivery. And this is a brilliant approach. I absolutely love it. And I hope to see something more done because it's great. And I think it has a high potential. So this approach exploits the ability of reactive oxygen species to break membranes to liberate, to free drugs from lysosomal entrapment. So say you've got a drug that gets taken up by the cell by endocytosis. It ends up in the endosome becomes a lysosome, and the drug gets destroyed before it ever has the chance to go out in the cytoplasm and interact with its target. Now, if you treat the cell together with the drug, with the photosensitizer that is able to accumulate in the lysosomal membrane, as you can see here, once you have the drug in the endosome lysosome, if you deliver a little bit of light, you burst the membrane of the endosome, the drug is free, it goes to the target, and the drug action is achieved. This approach was pioneered by a group in Norway, Christian Berg's group, and it has been used by other people as well, and it proved quite useful to deliver drugs like doxorubicin, genes, vaccine, metabolic toxin, you name it. So drugs, and that sometimes you need to increase the dose to reach your expected therapeutic effect. But by increasing the dose, because you want to circumvent this lysosomal disruption, you start seeing toxic effects. So it's improving the delivery. This approach has gone through a clinical trial a few years back to deliver bleomycin for head and neck cancer, and it was proven, it was the first clinical trial whose aim was to prove that the treatment was safe and tolerable, which apparently was. What did we do with that? Now this is an old friend. We saw this guy so many times. That, you know, so it's our conjugate, usual conjugate. I made a lot, so we were trying to use it for as many things as we could. Here you can see this is a bright field image and punctuate distribution in the cell. So lysosomal entrapment, which we proved already. If you irradiate with a little light, you can see that the conjugate relocates and its distribution is more diffuse in the cytoplasm, meaning that this guy goes in the lysosome, and if you hit with a little bit of light, it goes everywhere. Okay, so this again is to show that the same distribution of the conjugate and the lyso tracker that shows where the lysosomes are, and the kind of merged images on HN5 cells. What did we do with these? We co-administered these with saccharin. Saccharin is a metabolic toxin, so it's one of the compounds that cannot be used as drugs because it gets destroyed in the lysosome, and if you increase the dose to see the effect, you kill everything, so it's not good. This is a busy graph, so bear with me. Purple and orange lines here, these two here, they are the cells treated with saccharin only, viability almost not changed, different concentration of conjugate, so porphyrin and a little bit of light. So if you increase the dose of light, like in this light colored light blue line, you start to see a little bit of toxicity, but still have quite a lot of cells viable. If you do the two things together, so if you administer saccharin and then the middle dose, the one that by itself doesn't do much to the cell, together with the saccharin and give light, then you have a drastic reduction of the cell viability, meaning that your saccharin is free to go and wreak havoc everywhere in the cell. Beautiful conjugate, this one. Right, last thing I wanted to talk about, I promised you drug delivery, drug formulation, and this is it. And I call it my curse because my office mate, who's not here today, otherwise she would have heard the whole story, is a formulation scientist. And every time I'm excited because oh, look, I made this porphyrin, I make this conjugate. I said, that's nice, but how are you going to put it in the cell? How are you going to deliver it? So, once we had a look at this, we selected, this is not a peptide for the moment. We're working with silk protein. We chose a protein that can be derived from silk as a vehicle for formulating porphyrins. Why silk fibrin in particular? Why silk fibrin? Because its history as a high potential material for medicine has been proven already, so we're not discovering anything here. It's biocompatible, biodegradable. It doesn't impair or sustains the cell proliferation. So it's a good material to use for regenerative medicine. And from the material point of view, it can be fabricated in pretty much any shape and consistency you want, with limits, of course. So this is a bit of what have been done, particles, spheres, films, fibers, the lot has been done with this protein. And the structure of the protein is that it is what explains its versatility, if you like. The silk fibrin has domains which are no water soluble that tend to stack with one another, but then there are parts of the chain which is water soluble, so it can absorb water but without dissolving in water. So it's got a lot of positives to be a material to support tissue regeneration and cell proliferation. What did we do with that? Okay, we optimized, we adopted an approach to extract silk fibrin from cocoons. You basically first you clean the silk cocoon with sodium carbonate, then disrupt the protein with lithium chloride and then dialyze, and you obtain a kind of a thickish viscous solution, which if you add alcohol, ethanol, it becomes a self-supporting gel. We took this gel and added different concentration of this particular porphyrin, which is water soluble, but it aggregates like it had nothing better to do in its life. And here you see the self-supporting gels with the different concentration of porphyrin, least concentrated, highest concentration. The fact that you don't see it shining here means that the concentration is too high, and these rings are too close to one another, they start to quench each other, okay? And what can we see here? For example, we can see that introducing the porphyrin in the gel is a good way to prevent, up to a certain point, aggregation, because if you look at this graph, it's quite busy, but the red line, which is this one, it doesn't emit a lot, or it emits a lot less than this line that I'm pointing at now. So in solution, these solid lines represent the emission in solution. The emission is quenched more than it is quenched when the porphyrin is in the gel. So it maintains a little bit more the emissivity of the porphyrin. This translates into a better generation of reactive oxygen species. It's okay. Reactive oxygen species, they're not easy to see because they kind of die in your hands when you make them. One way of seeing them is to trap them with fluorophores. And this is called single oxygen sensor green. You can buy it for quite a price, but it's non-fluorescent until it reacts with singlet oxygen, and in that case, it starts emitting green. Now, in this graph that I'm showing you here is the intensity of emission versus the exposure time of the porphyrin in water. So yes, you have production of reactive oxygen species, but you see a lot more fluorescence, hence a lot more of emission of reactive oxygen species if the porphyrin is in the hydrogel. And if you consider that the diffusion in the gel is not that great, it means that the porphyrin really works better in these conditions. So it's good to use proteins or peptides as well for the delivery of these species. Right, that's the end for me. So I promised the Mitral Alliance. Is it a Mitral Alliance? In my humble view, yes, it is. Once you can take non-water soluble porphyrins and make them water soluble by choosing the right peptide. You can choose the right peptide, and the porphyrin that wouldn't get into the cell gets into the cell, so you can increase cell trafficking. You can prevent porphyrins from aggregating in water, which again is good because it preserves and it improves the photodynamic performance. You can use it as a formulation trick, not just as a delivery trick. And let me tell you that from the point of view of a medicinal chemist, this is paradise because it's a lot of fun, and I want to do more and more and more. Last thing I wanted to do is to thank everybody that has been involved more or less with this work, starting from back when I was in Bath. So the conjugates with the cell-penetrating peptide, the people in Bath and the UCL in London, the cyclic conjugate, Paolo Giuntini, that just left my lab. He's finishing his PhD, Chris Coxon, who's now is in Edinburgh, and Keind and Talat here, silk fibrin with my collaborators in Brazil, and many more people that have, look, these two, you know. Yeah, so I swear I didn't prepare these today, they were here already, okay? And of course, these people for paying for everything I've been able to do, and of course, not least you for listening to me. Thank you very much.
Thank you very much.
Thank you, Francesca. It's a very nice presentation. There is time for a question. Okay. Sonia?
Va
Sorry.
bene italiano, in English, Italian, you do whatever you want. Mi va bene qualsiasi
For
cosa.
me, it's okay.
Okay.
What you want. If you want in Italian or in English,
right,
for
yeah.
me, it's okay.
Okay.
So I have a curiosity. Thank
Yes.
you very much for this nice presentation. Really, I'm a medicinal chemist, so I appreciate so much the chemical part. I have only a curiosity concerning the stability of the linear peptides in vivo. Since we know that normally they are not so stable, have you done some experiments in vivo or concerning the stability, enzymatic stability of these peptides?
Not directly. We know that they live as long as we need them to live, but there's a lot that can be done. Like, for example, if you replace, say, if you have a 13 long sequence, if you replace a couple of amino acids with a D amino acid, if it's a cell-penetrating peptide, it will keep on doing its job.
Okay.
But then all of a sudden it's not recognized by the peptidases and it lives longer. Or stapling, for example, the work I was showing where we put the peptide around the porphyrin.
Yeah, the cyclic,
Yes. But we haven't looked at it directly. Actually, I was thinking today that we really should have done it for the cyclic. Well, I'll ask my colleagues if they want to do it, yes.
Okay, thank you very much.
Welcome.
Okay, another question from Pietro Amico.
Yeah, good evening. Many compliments for the presentation. I am a PhD student. I will start my PhD within this year in Messina. Interestingly, your presentation, kind of things was involved in conjugation between porphyrin and natural polymer, which is part of my research project. So what I was wondering is that, would it be useful to conjugate the porphyrin with a natural polymer such as hyaluronan or compounds like that, trying to exploit the possibility to a targeted delivery, such as in cancer? There are kind of polymer that can be used as a delivery system to target the disease in order to exploit the features of the porphyrin. I don't know if you get what I mean.
Absolutely. I never the only natural, well, if you call it, that's another protein actually. I conjugated porphyrins on collagen and hyaluronan, not yet. What did I do? Starch. So yes, it's something. It's a field where there's a lot that is being done, but it's absolutely it's great. Yes, it's a good idea. I think.
What I was thinking was that in order also to improve the technologic feature of the porphyrin, in order to improve the water solubility, trying to conjugate a porphyrin with a natural polymer such as hyaluronan or other compounds which is similar to hyaluronan, could it be an improvement to the characteristic of the porphyrin on compared related to porphyrin?
I think so, because you can, it's a bit like the approach that you use on things like albumin. You can deliver a lot more porphyrin with just one nanoparticle. So the hyaluronan, I'm not sure, but other similar polymers have been used to make nanoparticles with porphyrins or other photosensitizers. So yes, I think it's a good idea. I'd have a go if I were you.
Is the photo activation maintained even if you use a conjugate like that?
I think the evidences that are out there suggest that yes, it's quite likely. Of course, every time you have then to test, but from what people have been doing, I couldn't see any reason to say this particular polymer will quench the porphyrin. Something like hyaluronan, I don't think it would quench the porphyrin unless you start putting too much and you have a little bit like we were observing in the silk. So if you overload it, then you have the rings that are too close, and then probably they won't do anymore, but they would, they wouldn't be efficient anymore. But chemically, the polymer, I don't think it would do any harm to the photodynamic efficiency of the porphyrin.
Greetings again. Many thanks.
Yeah.
Other question?
I have a question.
Okay.
I have a question.
Okay.
Francesca.
Nino.
How you performed the singlet oxygen experiment when you have a porphyrin hydrogel? Because you use I think is not soluble, not water soluble.
In water, it's not water soluble. So okay, so if you're doing on a gel in a material, even non-water soluble probes might become soluble. So in our case, we were lucky. And then in our case, a percentage of the continuous phase is ethanol. So I guess a mixture of the presence of a bit of a percentage of ethanol in the gel plus the presence of the silk, it helped bring in the species in solution, the probe in solution. The other way would do to attach the probe to a nanoparticle. Polyamide nanoparticle then will help suspending your probe in water, and then you have a water soluble probe. So it is dispersed molecularly sorting.
Okay, and the spectroscopic properties of the porphyrins inside the hydrogel, I mean the emission, fluorescence, and absorption is is different with respect to the porphyrin,
In
for example, in
solution?
water. Yes, in solution in water.
Not really. For us, it was better because they were less prone to aggregation. And if you have something,
So you study by diluting the
yes,
systems.
yes.
Yes.
At some point, you start seeing quenching, and then at that point, you stop seeing reactive oxygen species being produced as well. And I think you can load the gel a bit more than you would be able to load the solution, if you know what I mean. So you can get a more concentrated by volume, concentrated if it's in the gel than if it's in solution.
Okay, thank you very much.
You're welcome.
Okay, there is a question from Greta.
Hold on. Okay.
Ciao,
Ciao, Francesca. How are you?
Good.
Good. Very nice presentation. Thank you very much. Just a curiosity. I was wondering, maybe you mentioned, but I skipped it, but why did you choose to use silk fibrin?
It's a convoluted story. So one thing was that we wanted to look at spinning, electrospinning silk. And
which is very good, by the way.
Yes, and which we ended up by not doing for this project. And then when I was visiting Brazil, my collaborator, the collaborator I was seeing at the time, had a project that was concerning silk fibrin. So let's say that the meeting between me and silk fibrin was totally random.
Okay.
But then I really like it as a material.
Yeah, it's a very nice material,
yeah.
absolutely, especially for making, I don't know, patches or that kind of application.
Yeah.
So thank you very much.
You're very welcome. Welcome.
Bye.
Bye.
Other question? I have a question for you, Francesca. I don't understand exactly the role of the peptide chain in the active antibacterial porphyrin. Because in the structure of the porphyrin, you see that the porphyrin have the cationic charge. So the cationic charge have to attach with the membrane of the bacteria. So I don't understand exactly the role of the
peptide. So no, the porphyrin that I was showing with the one that we did is not positively charged. It's this one here. Let me see. No, not this one. This one. This is phenol. So this porphyrin is a piece of brick. And you see that?
No.
No, eccolo, vedi? Wait then. Talking about, of course, if I don't do, now you see.
No, before?
Before?
The slide before, I think.
This?
No, after.
I see. Okay, that is not my work. But, so this one here.
Okay, here.
Okay, so if you go and see this paper, you see that the porphyrin itself is cationic, and it does do a damage to the bacteria, but when it is attached to polymyxin B, it's a lot more.
Okay.
So in this case, it's like a synergy, if you like, because these can be antibacterial and photo-antibacterial. This is an antibacterial, so you need a lot less of the conjugate than you need of the two.
You attach the antibacterial to obtain
so this is not us. But so people normally they saw that if you use a peptide that is known to have an antibacterial property, you reduce the dose, you make a much better antibacterial agent rather than, because this is the same. So this is cationic. Okay, they did it with non-cationic as well, but this is a cationic. There is a cationic derivative. But when you have these peptides, it becomes like literally a bomb.
Okay.
So, yeah.
Okay. Thank you. Other question? Greta, do you have another question? Or
is it the hand from before?
Yeah, it's my hand from before.
I always forget the hand as well.
Matteo Di Giosia wants to make a question, I guess. Okay.
Yes. Yeah. Thank you for the very interesting presentation. I have just a curiosity about the stability of the peptide during the irradiation. It is known that amino acids are targets for reactive oxygen species.
Yes.
My question is, did you check if you have any change in chemical structure of the peptide during after the irradiation?
Yeah, yes, we did. We didn't do a systematic test, so I can't tell you how long the peptide will resist, but with the radiation time that we tried, the mass of the conjugate after radiation didn't change.
Okay.
So my wild guess is that we didn't have any residue that would really be prone to. So we didn't have histidine, we didn't have tryptophan, we didn't have tyrosine, I think, or thereabouts. So, you know, maybe if you manage to dodge the residues that are targeted more by the reactive oxygen species, maybe then you know, okay, this is a stupid answer. It's an answer I never gave. But if my peptide makes the porphyrin go where I want, then it can die for what I care. But this is not a good answer. So
well, it was just a curiosity if you have a strategy to check the stability of the.
We saw, as I said, we didn't do any systematic study, but the little bit that we saw, it didn't. The peptide was integer at the end. We did it with the TAT.
Thanks a lot.
Greta?
È sempre quella di prima, Enrico.
La mano di prima?
Che non la togli, pensavo che l'avessi, perché mi è sembrata che l'avessi tolta.
No, non riesco a toglierla io.
Ok, perfetto.
Ecco, ora l'ha tolta.
Okay, perfetto. Is there a question? Ok, no question. Ok. Passo all'italiano per salutarti.
Ma sì, ma grazie dell'invito, mi ha fatto piacerissimo. Mi avrebbe fatto piacere essere in una stanza con tutti voi.
È stato un vero piacere, assolutamente. Tra l'altro devo dire che mi ha riscosso anche un certo successo, perché vabbè, noi abbiamo il pugno del numero delle persone che hanno seguito, quindi
mammina, guarda quanto è stato... aiuto, ho paura, no? Via via. Boh, spero vi sia piaciuta, vi devo dire. Se avete curiosità dopo, googlatemi, e tanto la mia email esce fuori, quindi entrate in contatto pure, sono qua.
Ok, ricordiamo che comunque la presentazione è stata
googleremo.
Ti googleremo, sì.
La presentazione è stata registrata e quindi a breve poi verrà caricata sul sito della società. Come tutte le altre presentazioni, quindi uno magari ha modo di rivederla, anche di vedere tra l'altro anche i contatti di Francesca, quindi in questo caso poi di poter ha modo di contattare Francesca per ulteriori delucidazioni.
Va bene. Grazie mille ragazzi, davvero tantissimo.
Ciao Francesca, grazie.
Ciao, a presto, spero.
Ciao Francesca, grazie.
Ciao.
Ciao Francesca. Arrivederci, grazie.
Ciao a tutti.
Arrivederci, grazie.
Ciao.
Ciao.